this slide here is how we ended up, which is probably a slide you hate because it was not an original set for one thing. But I'm going to try to remember in a moment to start before I talk a couple of administrative things, including the error. Let me get the error dealt with first. This is this thing I sent out. And by the way, you probably noticed that I sent you invites to all of the, to everybody, just in case you want to review. But number 2, you should have known something was wrong here because it said life cycle with 1 kinds. And then the syntax was wrong. So and Steve sent me a reminder that it was 2. And that, by the way, it brings me to another thought. On my YouTube channel, there is the public thing is the Aster workshop. And the other public thing is a little tutorial that Steve developed about that time that showed you how to make changes to a PDF. And Steve, do you remember if that had, you may have to unmute to answer, but do you remember if that had audio? Because when I looked at it yesterday, it didn't. Okay, good. Well, I wasn't goofy then. But anyway, it does, and I have to, you can make changes to a PDF with that, if you look at that, if you don't know how, because I forget from time to time, it's kind of hidden. And because they want you to buy the Adobe version, and that's why they make it hard to get. But you can actually make, you can go in and make this 201 if you look at Steve's video. And if you can't find it, let me know, and Steve or I'll send you the link. But all right, now the other thing I want to point out on this thing is I'll be going over this. Well, this is the definition that I put forth about 40 minutes and 2 seconds into it. It's a very long definition. We'll be going over that today, again, because it's necessary to understand that part, that hallmark of sexual reproduction in order to understand genetics. And so that'll come in handy today. So you might have it handy because we'll be looking at it in slide form. Okay, let me X out of this if I can. Let me see if I can close that. Close that window. Okay, now as far as other administrative things, is anybody, let's see, did Amy ever come in? I don't see her. Anyway, geez, I think I could tell here if she was in or trying to come in. I should have it to allow her to come in automatically. I wonder if I can do that. Let's see. No. It should be an automatic invite. Anyway, all right. Now let's get out of that. Click somewhere else. Now, okay. I think I'll go ahead and, well, I'm afraid to minimize it because I might miss her coming in. But anyhow, next, has anybody got any reason we can't do it next week, Stu? Because I figure we got 2, maybe 3 more, then we'll be done. So it's a good day to be inside because August can be beastly, and July, the last 2 days in July was pretty rough too. All right, let's start with this concept because it's not a concept. Actually, I think it was on that PDF I just sent you, the term thallus. If it wasn't on it, it should have been on it. A thallus can be, well, first of all, it's a non-vascular plant body. And a thallus can be as simple as this adult chlamydomonas cell. It can be, let me make it bigger. It can be as simple as this one cell, a thallus. But when we think of thallus, it can be a filament. That's what happens when, let's look at this moss thing. When one cell settles down and starts growing, loses its flagella and starts growing, it becomes an adult. This is a gametophyte, the adult. I wish they wouldn't put GM here because it makes you think of gamete. It's germinating spores, what it is. And so this is a filament, and in an alga, that might be the adult. That might be all you get. But in the bryophyte, it becomes a little bud here, and then and then germinates fully and develops into a gametified, a haploid entity. And but anyway, we'll actually be talking about the difference between the products between these spores, myospores, and this and the gametes that do the uniting to make the zygote. This was down here later. So let me go ahead and exit out. I haven't seen Amy come in, so I guess she's not going to be with us. But let me close that window. I thought I was going to close it. Let's see. Okay, I'm going to minimize this, and I hope it means that. Let me change it. That makes anyway. All right, so that's why I started with that, because I figured the term thallus may not be clear, and we'll be revisiting this concept of how you tell us. I mean, the spore versus the gamete concept in a minute, because in genetics they skip something, and because it's easier to do that. Now this is the opening slide, and you might be going to be a little mad with me because you see this graphic over here on the right that's not on your slide, and the stuff in red is not on your slide either. Okay, so you might want to do that. And this actually is the same. I just took this. Wait a minute. I need to be. Let me do my little annotate thing here. All right, now. Let me move that up out of the way because you can't see it, but it gets in my way. All right, now I pasted that excerpt into this slide, and I realize that there's some modifications that weren't on your copy, and so I'm giving you time to add them in because the stuff in red shouldn't be on your slide, including this drawing. I knew the drawing wasn't on it, but this morning I realized that this verbiage, this addendum wasn't on there. And this wording goes along with this thing. Let me, Steve, you got any idea whether I could see Amy or not in this view? I don't think I could, but. Okay, all right. Let's see. Let me go back to the gallery. Grid view might be good. No, I don't see her. Let's see. Yeah, I don't see her. So I guess I'll quit worrying about it. And it is what it is. All right. Okay. So all right. What I was trying to do here was rehashing the significance of meiosis. Everybody knows about the fact that meiosis brings the chromosome number back down to 23 in humans. If it didn't do that, then it would go to 96 and 92 and so on. And in garden peas, which is Mendel's classic organism of plant of choice, it's diploid and haploid, 14 and 7 in that order. Now, the second part of meiosis is the genetic significance of it, and it's besides the mathematics of the numbers being halved, there's the genetic crossing over that can take place at metaphase 1 of meiosis. Now, the diagrams I've been showing you, this is a tetrad of homologs. These two are from one parent, the two in the background are from the other parent, maternal or paternal or vice versa. And when they're lined up on the equatorial plate of my starting to before they separate, there's an opportunity for exchanges of DNA to take place, and there could be as many as 50 genes going back and forth between the parents, homologs, and that's an opportunity. Well, it could be more than that. I just threw out that as this little squiggle here could represent 50 genes, and this 50 or more, 100. And so it's kind of like it's like shuffling a genetic deck. And that's why your siblings don't look alike and so forth. That's one of the reasons. All right. So that is a real significance of meiosis. Two things, two aspects. Now, this is where that long definition might come in handy, because I want to point out the differences in meiosis and mitosis. And we're winding up plant reproduction with this being a prelude in the genetics. Mitosis can take place in a haploid cell or a diploid cell. All right, so I already X'd out of it, but all right, that spore that I showed you that germinated into a filament is haploid, and it's undergoing mitosis to form the filament. And then the filament continues to form a bud and then a moss plant. So all that indicates that a haploid cell can do mitosis, and it does. In humans, though, your haploid cell gamete doesn't. It dies or unites, and that's the difference in a spore and a gamete. Now, meiosis can only take place, though, in diploid cells. You can't halve what's already been halved. You can't, the 23, once you get to that 23, you can't bring it. Well, first of all, that's an odd number, you know, and you couldn't divide 7 to get a whole number. All right, so those are differences already. Now, this is a very important difference between mitosis and meiosis. The members of the homologous pair, these don't line up at metaphase 1, or the only metaphase there is in mitosis. Over here, they line up. That lineup is not very well depicted here. It's more like, I'm having this. Let me do this. It's more like this kind of lineup instead of this way. The orientation of the members of the homologous pair is side by side. Truthfully, it's more like this. And don't worry about these, all of this going on, but this is the spindle apparatus that's present that holds things in place on the equatorial plate in the middle of the cell. But the lineup is shown to be here, but better shown here. The truth is, but in fact, they're slam dunk right on top of one another. Okay, so okay, now, so that is a significant difference between, so the metaphase of mitosis, the only metaphase there is, and the metaphase 1 of meiosis is significantly different because they're lined up that way so there can be exchanges of genetic material. Over here, that's not going to happen. And another thing is the second part of the definition that I referred you to on the sheet, that Errata sheet, definition sheet, and Errata sheet says that meiosis consists of 2 successive nuclear divisions. That means one right behind the other. And then the second part of meiosis is mitosis. How can you say that? Well, because in that you separate chromatids, the double strands, you separate them. And that's what you do in all of mitosis, you separate the strands. There's only one duplication of DNA, and that takes place during interphase. In fact, technically, interphase is not a part of mitosis nor meiosis, but it is a necessary prelude. It's a necessary prelude to both of those processes. And that it's no accident that this represents a big chunk of the slide, because interphase, although it's not a part of mitosis, it's a necessary prelude, because that's the interphase is the stage where the DNA replication takes place. I'm going to say 2 things about it. Interphase used to be dumbly referred to as the resting phase. Well, it's anything but resting, because if you're replicating DNA, you ain't rested, folks. You're making you're replicating this strand into its duplicate. And at this point, the chromosome consists of its sister chromatids, and here it's not. Another thing that I've already mentioned, and I'm going to mention it again right now, is the fact that the chromosome is not visible like you see it here during interphase. And in order to understand that, think about the difference in a piece of macaroni, like you see here, and a piece of spaghetti, like you don't see. The spaghetti is a long strand of spread out DNA. That's how it looks during interphase. You don't see a chromosome well defined during interphase. It's like a tremendously long strand of spaghetti, and that's where the DNA replication or duplication takes place. So don't get the idea that during interphase the chromosomes look like this. They don't. All you could see is a bunch of chromatin dots, and that is 90% of the cycle of the cell. The division is only 10% of the cycle of the cell. And sometimes we'll talk about mitosis, meiosis, and I think it's, let's see. Let's see. Haploid cells with unreplicated chromosomes is a product. Now, I already talked about this. All right. Let's see. Where was it? Okay, here. All right. The 2 diploid daughter cells. Here is talk about 4 daughter cells. That's another difference, 2 and 4. But let me make something clear. It's really the definition of both of these technically is daughter nuclei, not cells. Cytokinesis takes place in both. Cytoplasmic division accompanies both of those. Okay. Now for genetics. If you haven't figured it out already, you're going to figure out that plant reproduction is a necessary precursor to plant genetics. And the next topic, which we might not entirely finish this today, we'll see. The lead into plant evolution is plant genetic. And so there's, and I don't think I saw those connections back when I was first getting introduced to this. Now, I was going to ask you the question when you had your audio on, and I'm not going to, how many of you, if any, have had a course in genetics? All right, okay. I'm not hearing anybody, and it's not surprising that you haven't, because most of you probably don't have degrees in biology, and I've known of at least one good botanist that doesn't have, that never took a course in genetics, which I can't understand how they could get away with that. But kind of like your parents, your advisor will make you do stuff you don't want to do, and later on you'll see the advantage of it. But somehow or another that person escaped that, and I'm sorry for them, because it's such an eye-opener. It's one of my favorite courses. I was taught genetics by an entomologist, a bug guy. Especially, he was dragonfly expert. But anyhow, he did a good job, and he probably dealt more with fruit flies than he did plants, unlike Mendel. But when he taught it, but that was okay. Now, so we're going to go over some introduction basic terms in genetics that are necessary in the first part, especially for those that haven't, and nobody's had genetics apparently. We'll go through the classical or Mendelian genetics, and then we'll summarize the big 3 laws: dominant, segregation, independent assortment that Mendel came up with. Came up with some other stuff that's not in his laws, and that's what this is about. We'll be lucky to get to the end of that, but today, because I've already gone 20 minutes. Now, and then we'll talk about beyond Mendelian genetics, some things that he didn't know about that we've later learned. And then the wonderful modern synthesis, that's what happens when genetics meets Darwin's evolution, and the population genetics went on in the 1930s and so forth. And so it's the putting together or compounding of all of what we knew into the modern synthesis of evolution, genetics, botany, zoology, ecology, all that gets lumped into, and genetics becomes what it is today, the modern synthesis. Any questions? All right, now, besides the ability of living organisms to reproduce more of their own kind, another distinguishing trait of living things is their ability to transmit traits from one generation to the next. If I was introducing a basic biology class, I would say that biology is a study of life. But at the same time, I would say it's easy to define biology. It's not easy to define what life is. And then I would go into my spiel about the traits, and one of them would be living things can transmit information from one generation to the next. And that brings me to a very relevant point that's going on today, that you could argue that viruses aren't living things, because viruses replicate only when they're in living things. They're parasites, and that's what one of the things that parasites do. And they might be tiny, but you can't argue that they are a heavy-duty prize fighter when they can bring down a human. So little things without brains aren't necessarily dumb just because they don't have a brain. When something like that can affect the human species to the extent that it does, it's not insignificant just because it's small. And people tend to think, it's humans and animals. No, it's humans and other animals, because just because something is not a human doesn't mean it's not important. And that's where I get in trouble with religion. I really have a big bunch of bones to pick about that. But so much for that tangent, but it's related to COVID too, because, all right, now, the ability to do what's set up here in this book is called heredity or inheritance. And the offspring differ somewhat from the parent and sibling, and that's demonstrating variation. And genetics is a study of heredity and variation. And that last word, variation, is something that should ring a ding bell for you to think about evolution. So, because that's a natural lead-in into evolution, okay? One could argue that variation belongs in evolution, which we have indirectly incorporated from time to time, and as we will today. But we'll be bona fide into evolution next time, or certainly the time thereafter. Okay, I think that does it. Now, this is a busy slide. It's a slide that are going to violate the rule of slides and probably stay on longer than a minute or so, but because it's got so much information in it. All right, the protein part of a chromosome is, all right, first of all, a self-replicating cellular structure composed of DNA and protein, by which hereditary information is transmitted one generation next. The protein part goes by the term histone, Histone. Now, Donna and others, I'm not going to keep bringing in terms, too many of them, I don't think, but that's not a real mainline important term, but that is what the protein is. It's called histone, and it's the protein that's most responsible for the chromosome's ability to become compacted into a small space, like inside the nucleus, along with all the other chromosomes. That's one of the big roles about histone. Now, the purist would add to the definition that I have here that the DNA also includes small bits of RNA, but into the mix. But that can be confusing to do that, so we're going to leave it out. But it's the truth, though. Purists could argue that. Now, as I've already said, chromosomes become recognizable during cell division. They become shortened and thickened and contracted. They become macaroni instead of chromosomes. Now, the gene is that part of a DNA molecule necessary to code for a particular kind of protein, or in some cases, other worker molecule like RNA. So, and this is a representation of what a graphic that tries to illustrate what a gene is. There's your DNA double helix. Don't worry about these terms, intron and exon, because they're not really important for us, but I can explain at the end for you if you want to stay on. I can explain for you what they are. But I will go ahead and say that an intron is spliced out, removed, when the DNA is transcribed into RNA. And because in order to make a protein, you have to involve RNA, the other kind of nucleic acid, which is also, like DNA, made up of building blocks called nucleotides. Now, what's the building block for a protein? Amino acids. The building block for a DNA molecule is they're made up of nucleotides. And this building block has three components. It has a phosphate group, a 5-carbon sugar, either ribose, like here, or deoxyribose in DNA. So this would actually be a nucleotide of RNA because it's got ribose instead of deoxyribose, which has one fewer oxygen. And then there's something here that's a heterocyclic nitrogenous compound called a nitrogenous base, and this one happens to be adenine. There are 4 nitrogenous bases involved in nucleic acids: adenine, thymine, guanine, and cytosine. They pair up. Now they pair up here in DNA molecules. Think of this crossbar. Okay, I'm not a big football fan anymore much, but think about the goalpost. This is the goalpost, the line that goes across. And the goalpost that goes up in the air is represented by that segment and that segment. The part that goes below the ground is represented by this segment and this segment. So think of a distorted H. And every H every H in the crossbar. All right, this is primarily of adenine and thymine, or guanine and cytosine. This is at the junction of these, where the red meets the walls. And the walls are where the phosphate molecules and the sugars come in. So what I'm trying to tell you is the nucleotide, this building block, is incorporated into this DNA helix. Now, a gene, I've already given you a simple definition, but let me talk about an example of it. I used to tell people that insulin was made up of about 75 amino acids. It's more like, today we know it's more like 84 amino acids. And insulin is a relatively simple protein made up of as 2 polypeptides. One of them has 51 nucleotides in it, 51 of these, and the other has 31. Okay, so there's 84 amino acids involved in the thing, but 153 different nucleotides get involved in the process. Okay, so and a mutation could be, when we talk about evolution, we'll talk about the fact that a simple mutation could be anything that's as simple as replacing one amino acid, which is what we'll get in sickle cell anemia. All right, I'm sure I lost you now, so proteins are enzymes, among other things. They can be structural, but they're enzymes. And when the worker molecule is a protein, it's usually an enzyme that makes something happen. All right, enzyme sucrase, S-U-C-R-A-S-E. There's a DNA molecule that's responsible for making that enzyme, that protein called sucrase. And by the name sucrase, that enzyme must be instrumental in making sucrose. Taking glucose molecule and a galactose molecule and putting them together, those 2 monosaccharides, to make a disaccharide called sucrose. And that's so a DNA molecule that makes sucrase is an important DNA molecule, okay, because it makes things happen. In other words, so the DNA molecule is a blueprint for making protein. That's all it is, a blueprint for making proteins. And I've already said the blueprint for making insulin consist of 153 of these nucleotides, not all the same, but different in different combinations. Now, this stylistic diagram over here showing a gene is showing it in relation to the double helix. And the chromosome is X-shaped during when it starts to divide. This segment, this diagram labels a region of only about 50 or so nucleotides. Okay. In reality, most genes are hundreds of these, hundreds of nucleotides, not just a few that you see here. Okay, there's probably no more difficult slide than this. If there's any questions, let's try to get them out of the way now, because I know I've lost some of you on this because I tied a lot together and I stayed on the slide very long time. Anybody got any groundbreaking questions? All right, hearing none, we'll move on. Now, all right, the terms themselves. An allele, that'll become more obvious in the next slide or so, but it's an alternate form of gene, like purple and white would be different forms of color. A locus is the place on a chromosome where a gene appears, okay? And that term will become a little more apparent too. Chromatid is a term we've introduced already. You don't have a chromatid unless DNA replication or duplication has taken place. And the homologous pair consists of 4 strands in the homologous pair, 2 strands on one chromosome, paternal, 2 strands on a maternal chromosome. No. You don't have that until replication takes place. Phenotype is more or less in fact, the word is Greek for appearance, but it's a little misleading. It doesn't have to be appearance. It can be a chemical, it's more or less morphology, but when bacteria, it can be something as simple as biochemical, physiological, like some bacteria would have an enzyme that makes certain things happen and another one would not. A bacterium that had an enzyme called urease could break down urea. Most bacteria don't have it, but it can be as simple as that. And that kind of difference is very important in identifying bacteria, more so than morphology, because let's face it, there aren't too many shapes of bacteria, but there's a heck of a lot of species. The species are told apart by the physiological, biochemical differences, most of them. It could be, and this is something that's hard to swallow, but I just said it could be appearance, but I brought about physiological changes. That could be non-appearance, but something as simple as animal behavior, like crickets having different mating behavior, that could be phenotype. Even though you can't see it, it's manifested, where you can tell the crickets apart by their mating behavior. So that would be phenotype, even though it's not morphology. Lactose intolerance or food allergies would be differences in phenotype, even though they're not appearance type things. And as the next slide will show, the genotype, we'll concentrate after this in trying to distinguish between genotype and phenotype. An organism's genetic makeup is the genotype, and the phenotype the genotype is not always obvious from the phenotype. That's the problem. It is obvious. The genotype and phenotype are really easy to tell and synchronize when we have this kind of condition, a true breeding situation, where the members of the homologous pair carry the same identical genes, not the alternate form here. So if the paternal and maternal chromosomes carry a pair of identical genes or alleles, then the phenotype is obvious. I mean, the genotype is obvious from the phenotype. If this was red and this was white, we could tell by saying, oh, the red one has genes made up like that, and the white one has genes that look like that, and we'd be correct. But by the way, I really hate the fact that, I'll bring it up in a minute about the symbolism associated with genes. In the right way and the wrong way. We're going to do the wrong way, unfortunately, because that's what the textbooks do. But heterozygous is when there's two different alleles for a character. Big R or little r, that would be heterozygous. A mongrel or monohybrid, a heterozygote is not a true breeder, because you breed, we're going to find out in a minute, you breed a big R, little r organism with another like organism, you're going to get something that's not breeding true. You breed that with two big R's with two big R parents, to have parents like that or like that, you're going to get offspring that are identical to the parents, and one that'll breed true. A homologous pair, we've already said, is those that pair off in metaphase one meiosis. We'll be seeing that again. And a mutation is something we'll concentrate more on when we get to evolution. But it's an inheritable change in the genetic DNA material of a cell. And that's, unfortunately, that's what's going on with the dadgum COVID right now. And the more opportunities, the more people that are unvaccinated, the more opportunities there is for mutations to take place, because they take place when the organism is going from one transmitted to one host to another. That's when it takes place. And so that's what we see, unfortunately, can't seem to get in the heads of these people that just don't, they're just too, I'm sorry, they're just too, well, if you don't know about something, you're ignorant, but if you know better and still do it, you're beyond that, you're stupid. Anyway, that's my favorite. Smoking is a good example, and texting while driving, which I'm stupid, once in a while I'll still do it, but it's a way to have an accident, I'm telling you. All right, now, you are what you are for two reasons, people: your genetic makeup and your environment. And because the genetic environment has an effect on, the environment has an effect on the genes. Two identical twins can be different because one smokes and one doesn't. That actually isn't, it's hard to think of it that way, but smoking is an environmental modification of those genes. Or secondhand smoke would be too, or pollution would be too. And so we're showing an acorn here with the genotype is shown here, the chromosome makeup, and then we're showing the phenotype as a seedling and then an adult. But and I can be in these hydrangeas, I always forget it. I think that the red color, it could be the pH of the soil. I believe this is acidic and this is basic, but don't quote me on that. But it can be as simple as that. The acidity of the soil changing the expression of genes, okay? So anyway, that's, you are what you are for your genetic makeup that you inherited, but what you ultimately become has a lot to do with the kind of environment you grow up in. This is a good example of phenotype being modified by an aquatic buttercup. A good number of aquatic plants have this tendency to have submerged leaves are dimorphic from the emergent leaves. And so these have genes in them that are identical to these genes in these cells of these leaves. But the microenvironment of the water versus the air can manifest the phenotype difference that you see here. One of the best ways to try to illustrate what you are and what you are because of your environment. So, now, the relationship between genotype, as I've already implied, is really simple. Furthermore, the phenotype can be used to refer to the appearance of an organism, overall appearance, due to, or it can be simply due to one character. Okay, well, I said that backwards. here the phenotype can be used to describe the overall aspects of the organism, but it can refer to one character. So it has a broad usage. Genotype can refer not only to a single genetic locus, that place on a chromosome, but to the organism's entire genetic makeup. However, the better term for the latter is genome. The entire genetic makeup of an organism on all 46 chromosomes is better referred to as the genome. In a human, it'd be 46. And an organism phenotype reflects its overall genotype and unique environmental history. Again, you are what you are for two reasons: your genotype and your exposure of your environmental history. Okay, old Gregor Mendel was an Austrian monk, and like most monks, the work ethic is strong. And he worked on his experiments for 7 years, and we'll talk about why it took 7 years. But he was an Austrian monk who studied the garden pea, studied patterns of inheritance in garden peas. Now, monks were big on gardening, so he was no exception. He did back then. They would call them hybridization experiments, and not genetic experiments. He was a contemporary, interestingly enough, of Darwin in the late 1850s and 60s, but they never knew each other. And after 7 years, he finally got around to publishing his works in an obscure journal in 1866. If you want to know, and you can Google it, but it's Proceedings of the Natural History of the Society of Brunn, B-R-U-N-N, with an umlaut over the U. And that was just buried for 35 years, and nobody, that journal was not a real popular journal. And it wasn't uncovered until the early 1900s. And then it was an eye-opener, because what he published back in 1866, if Darwin had known about it, it would have been a really good thing. But that's where we'll get into the modern synthesis, and when Darwin stuff unites with Mendel's stuff, and then all the other information, and that's where the modern synthesis came about. So the modern synthesis is an amalgam of both of their works, plus some other things like population genetics. But all right, now the father of genetics, he's called the father of genetics, Gregor Mendel is, because he really, gene wasn't even a word he used. He called them factors. But so the word came about later. But he selected the garden peas. Like I said, the monks of the monastery had a long tradition of interbreeding plants, in this case, peas. Mendel luckily selected garden pea, and he was lucky in the traits he chose too. It has several distinct heritable features, characters with different variants, and we're going to go over those 7 that he chose in a moment. Another advantage of the garden pea is that they self-pollinate, and he can cut off the stamens and control which plants mate with which. He could also transfer pollen from one plant to another. He could cross-pollinate. So those are the kind of activities. Obviously, he would do his emasculation before the pollen was spread. Okay. In order to do transpollination, though, some of the pollen had to mature. Now, these are the 7 traits he chose in garden peas. Now, we're going to talk about later. I got a little boo-boo in the slide. Let's see, I think it's about 2 or 3 slides away. Smooth is the same as round, okay, phenotype-wise, and it's not important now, but it'll be important when I show you the boo-boo on the slide that I discovered Saturday or Friday. I don't think we're going to finish the day at this rate, but that's probably a good thing. But because this is so if you haven't had genetics, this could be rough. But anyway, all right, the alternative is wrinkle, smooth and wrinkle seed texture. Seed colors number 2, yellow or green, purple and white, and then axial flowers versus terminal flowers is actually this flower color is going. We'll talk about it next time if we don't get into today. That there's a link. There's connection between flower color and flower position. It's called pleiotropy. So don't write it down now. We'll bring it up later. Stem length, pod shape, and pod color are the total number of 7 traits that he picked. Looking at only one of these traits, the one I've got the arrow at, flower color, we come up with this situation. This actually explains the locus. There's a place on a chromosome where a gene appears, and there's different alleles here. There's an allele for purple and versus allele for white. The symbolism, let me go ahead and spill the beans on that. The best symbolism would be to use W, because that way the recessive trait, the little w, gets not lost. But unfortunately, what we're going to do is what textbooks do and use P. The P, big P is purple and little p is unfortunately white, as opposed to big W and little w, which is what the symbolism really ought to be. So, so be aware of that problem. Mendel was lucky in that he chose not only, let's see, well, his laws of independent assortment would not have worked if he hadn't chose traits that are located on different chromosomes too. And that's really something. All 7 traits were on different chromosomes. And there's only 7 kinds of chromosomes in P anyhow. So how lucky is that? He was also lucky that he chose that traits were all they were discontinuous traits. In other words, the flower color was either purple or white, not pink, not blended inheritance. So incomplete dominance would be if they were pink. So he was lucky in that respect. Okay, now still looking at only flower color. Here we go. Purple and white. Now, typical breeding experiment like that seen in the next slide here. That might be Richard Ware. Richard Ware just put out the plant contest. Today is the 1st of August. And since this is recorded, I'm not going to elaborate, but basically I would be participating in that. And so I'm sacrificing winning it for this, but that's fine. I don't need to win every time. Somebody else needs to win. And so hopefully someone else will submit their answers before I do. In the first 7 months, I've been first in the monthly competition, which is, one year I did all 12 months first, but I've never tried to concentrate again. I want to give other people a chance. Anyway, back to this. In a typical breeding experiment, like that seen in the next slide, Mendel would cross-pollinate, hybridize 2 contrasting true breeding varieties of peas. Now, okay, here we go. There's a true breeding with another true breeding, and then you're going to get a monohybrid offspring. There it is. That's a mongrel, heterozygote, because it's heterozygous for one pair of traits. And that's what a hybrid is. A hybrid is not going to breed true like the parents. That parent will breed true, and that one would too if they were crossed with if big P and big P were crossed, the 2 big P's were crossed with another big P, they'd breed true, and so would this one. But trying to breed these parents the offspring will be heterozygotes. It won't breed true. We'll see that in a moment. Okay, and I just said that. The true breeding parents are the P generation. That's the parents. And their hybrid offspring are the F1 or filial generation. That will not breed true. Mendel would then take the F1 hybrids, these guys, the the F1 hybrids here, he would take the F1 hybrids and allow them to self-pollinate and produce the F2 generation down here. It was mainly Mendel's quantitative analysis of the F2 plants that revealed the first two fundamental principles of heredity, the law of segregation and the law of independent assortment. And the truth is, to do independent assortment, you'd have to have the F2 generation. And that'll become obvious either late this time or the next time. In other words, you'd have to do a dihybrid experiment, not a monohybrid experiment like this, to see that the chromosomes assort independently. Now, you have to put that in your pipe and smoke it and think about it, but it won't become obvious immediately. This is what's known as a Punnett square. The Punnett square, P-U-N-N-E-T-T, it's better, here it is here. This is a Punnett square. I'm sorry, this is not. This Punnett square is the same thing as this slide, but it's in a way to understand it better. All right, this is trying to get you to understand what a monohybrid cross is. A cross of pure parents to yield a trait, offspring F1 that differs by one trait, monohybrid. And in this F2 generation, there's a 3-to-1 phenotypic ratio, red to white. Okay, now, and there's the numbers that turn out to be the 3-to-1 concept. All right, and the Punnett square predicts as the results of genetic cross between individuals of known genotype. The individuals of known genotype were these. Pure purple cross with pure white will yield a non-pure monohybrid. Cross self the monohybrids together and you get a dihybrid down here. Let's see if you see it. Okay, it's not obvious. All right, so, all right, in this dihybrid or F2 down here has a phenotypic ratio of, I thought it showed here. Where does it show? Okay, well, the dihybrid has a phenotypic ratio of 9 to 3 to 3 to 1. I'm getting ahead of myself. All right, look at this. All right, this has a genotypic ratio of 3 to typic ratio. Phenotypic is 3 to 1. Genotypic is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. 1 to 2 to 1 is the genotypic ratio. 3 to 1 is the phenotypic ratio in a dihybrid cross. Now, excuse me, this is still a monohybrid just to the F2 generation. All right, I'm sorry. Now, if Mendel, question is, if Mendel bred garden peas for 3 generations, why did it take 7 years to complete these experiments? The answer, Mendel had to be sure that he started his experiments with parents that bred true. All right. In other words, he had to start with parents that were like that parent and that parent. Okay, homozygous dominant for yellow, for smooth ones, homozygous dominant for wrinkled ones, homozygous dominant for yellow versus homozygous recessive for green. So he had to start with experiments with parents that bred that, that looked like that genotypically. And it took him 7 years to make sure he was dealing with the right parents. In other words, a more direct question of this first bullet here is, how was Mendel able to be sure of the genotypes of initial parents? How was he able to know that he had that if it was a monohybrid, or if he had the other trait, the shape, how did he know? And the answer, he had to do a test cross. And the test crossed has, something's not showing here. What is not? That's funny. Okay. All right. It's not possible to predict the genotype of an organism with a dominant phenotype. In other words, you can't look at a purple plant and tell if it's homozygous dominant or whether it's that heterozygous. But you just can't tell by looking at it. And breeding dogs is a good example. You can't tell by the looking at something what its genotype is. Now, the only time you can do that, and what you see is what you get, is if it's homozygous recessive. If an organism is homozygous recessive, you can tell that what you're looking at is the genetic makeup. This individual is white because it's homozygous recessive. Little p, it should be little w, but it's not. So a test cross is what Mendel did, a breeding experiment where he crossed the homozygous recessive, that, with a dominant phenotype, either that or that. If he crossed it with a dominant homozygous dominant, everybody would be purple. All the offspring would be purple. Big P would be, they'd all be heterozygotes. They'd all be purple, but if he crossed a heterozygote parent, a big P and little p, with a homozygous recessive here, half of them will be white, and half of them will be purple. That's in a monohybrid situation, a test cross, you're going to get that. Half the offspring purple and half offspring white. Not 3 to 1. Okay, not 3 to 1, like we got here. All right, now, all right, let's see. Mendel's law of segregation. Each pair of alleles separates during I think we'll go into this and we'll finish pretty quick here, but I want to try to make a point before we do. Each pair of alleles separates during meiosis so that the gametes, actually the myospores, receive one member of each pair of the alleles. Now, okay. In animals, it's easy to talk about this because the products of meiosis are gametes. In plants, the truth is, in short, that the two alleles separate into spores instead, ultimately gametes. Now, again, if we're talking animals, the product of meiosis is gametes. In plants, the product is spores. Ultimately, we'll deal with gametes. I've already thrown away that darn thing. Let me see. All right. Remember that moss, let's see if I can go capture that. I can't at this point. All right. Okay. Remember I started with I showed you that moss where the spores are germinating into filaments. That's a spore. And the spores grow into the filaments grow into gametophytes produce gametes. All right, so all of that goes on in here, and it's just not shown in plants. So the product, the P1 in a plant is really a myospore, not a gamete. But the myospore produces a gametophyte, it produces gametes. And I'm sorry for the way this is confusing, but to add all that in would make it more confusing. The bottom line is, after meiosis, you do not have both genes in the gamete or spore. You have a gene for a big S or a gene, in this case, in this parent, or a little s. All right? Again, and then the gametes have, comes in terms of the Y trait, it has one member of the homologous pair gene, big Y. This one has the other member of the homologous pair gene, the little y. When the P1s unite to form a zygote, you get a heterozygous zygote, one that is heterozygous for shape, heterozygous for color. This one appears smooth and yellow, but it carries genes for wrinkled and yellow. Excuse me, wrinkled and green. See why we need the different symbolism. Okay, the F1 gametes are as you see them here. Okay, now there's a typo that John pointed out. It's linked, not liked. And that's when he got the slides. Mendel's experiments. All right, the law of independent assortment. In order to get it, you had to be dealing with two traits, not one. So it wouldn't, a monocrybrid wouldn't do it. To validate the hypothesis is non-linked. You get the dihybrid cross, which is what we're going to go here. So he concluded that other experiments in which he followed the inheritance of two different characters are called a dihybrid cross. In this case, this is what the symbolism is. Big Y for yellow, little y for green, totally like backwards from what it should be. The textbooks do it, and unfortunately I'm doing the same thing. Yeah, okay, the allele for round is big R and little r instead of wrinkled and big W and little w. Now, Mendel's crossed a true breeding parents that had yellow round with the ones that were homozygous recessives that breeds true and producing something that's not on your slide, a dihybrid. So you need to write that in your slide. And the dihybrid, one possibility would be that the two characters are transmitted from one parent. All right, one possibility would be what you see here. And this, if you crossed, all right, this turned out to be an hypothesis that's not going to work for Mendel. It's dependent assortment. And the reason it's dependent assortment is the traits are linked on the same gene. And Mendel, as we said, was lucky that he picked traits that weren't linked on the same gene, because the traits, color and shape, can't assort independently. So you got dependent assortment in a linked gene situation. Now, this slide is the one I sent you lately. In other words, what you see what you see here, the linked genes, is this situation. The traits are linked, and they're not going to sort independently. To sort independently, the trait for color has to be on different homologs from the trait for shape, round versus wrinkle, yellow versus green, have to be on different chromosomes. Okay, and I'm going to stop there because this is, I can see already, even though I can't see you, I know your mind is twisted a little bit at this point, and I'm partly responsible because I didn't do as good a job as I thought I was going to do. But anyway, so in a dihybrid situation, if they're linked, you're not going to get a 9 to 3 to 3 to 1. You're going to get a 3 to 1, which is like a monohybrid cross. So this is not consistent with Mendel's results. To be consistent with Mendel's results, you'd have to have the traits on separate homologs. And then you'd get this, the 9 to 3 to 3 to 1 phenotypic ratio, and the 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1 genotypic ratio that I'm sorry I've said. All right, we'll cut it there. And I guess Amy never joined us, but maybe it was a good thing based on how this went. But all right, I'm going to back out of this and unshare the thing and stop the share. All right, has anybody got any questions? Yes. Yes. Not well. Allelopathy is when a set of a genetic makeup is responsible for usually some kind of poisoning effect, like a parent can kill its own offspring, or a species can chemically produce genetic the genes will produce poisons and proteins basically that kill other plants. But that's the connection. Allelopathy. It's a genetic poisoning going on, either your own offspring or others, usually others though. But creosote bush will keep its offspring from growing too close. The magnolias seem to have some chemicals that keep other things, including some grasses, from growing underneath them. But anyhow, so an allele is an alternate form of a gene, not the norm. Okay. Any other questions? All right. So probably a good thing we didn't finish, because this will give you a chance to write down some questions. And okay, I'm going to end it so we can cut the time frame. We'll pick up where we left off. It's probably a good time to break. Because we'll finish up with a better explanation of independent assortment, and then go into beyond Mendel, and then lead right into evolution. So I'll probably give you the evolution slides, even though we won't get very far into evolution. All right. Okay. I'm going to end it.